radical transformation. My name is Remina. I'm a research director working in the arts, and my background is in biology and computer programming. I lead R&D for artists who work with living materials. And just an example case study I've worked on are Annika Yee's kelp sculptures for the 2019 Venice Biennale.
My name is Shuyi. I'm an artist specializing in sculpture and installation. My interactive artworks focus on ecological themes and incorporate sound, electronics, video art, and biomaterials. Our practice as a collective is formed by 3 pillars: transdisciplinary research and development, artwork production for exhibition, and education. The experimental partnerships and collaborations we form through our work are a central part of our creative practice. We are currently producing our first large-scale solo exhibition in New York and looking for support with this project. As our collective debut, this exhibition contemplates decay as metabolism's twin shadow and explores the aesthetics and transformational capacity of decay.
For this exhibition, we are forging a research partnership with biotech company Kingdom Supercultures. We are working with the company founder to help him develop a sustainable model for art collaboration for the foreseeable future. And together we will develop a toolkit for other artists and scientists to follow as they try to initiate their own creative partnerships. To speak a little more on Kingdom Supercultures, they are a microecological design company, and they represent a radical shift away from biotech focused on chemical and synthetic food production. Both Decompose and Kingdom Supercultures are interested in the rich biodiversity of microbial life. Through our partnership, we will explore a library of thousands of microbes and study how these communities interact with each other in decay and fermentation. We believe the cultural taboo around decay prevents us from acknowledging a complete ecological life cycle. After all, for a microbe, a rotting apple is just a more efficient way of breaking down sugar. And we want to look at decay from all angles. That includes the algorithmic, digital decay, microbial interactions of decay, and ecological. The research we perform with Kingdom Supercultures will inform our resulting exhibition and educational programming.
Our video work is a meditation on decay and biotechnological attempts to optimize living materials. This video describes the human longing to control life and maximize productivity along multiple timescales and structures, from the molecular to the cellular, organismal, and societal. Here is a sketch of our work in progress, some design of our glass sculptures. Our multisensory sculpture installation will explore our concept of delicious decay through microbial communities. The living aspects of this series will be researched and produced through our collaboration with Kingdom Superculture. Data we collect from our microbial research, such as taste, smell, chemical composition, and micro interaction, will be analyzed with machine learning. Our microbe dataset will inform the design of our algorithmic sculptures. We will present these sculptures as a set of holograms that are subjected to the simulated force of decay. Compiling our research on decay, microbial ecology, and machine learning, we will publish a lexicon of shared language between art, science, and technology. This lexicon will be an educational tool for the exhibition and support our own ongoing curriculum development.
We can realize both the scientific residency and the exhibition at varying scales, and everything is in place, all the infrastructure and the partnerships for these 2 projects. But to make them fully viable, we need total financial support ranging from 15 to 30,000. And to this end, we are seeking financial and developmental partnerships, especially for our lab residency. And we are also seeking support for machine learning research and glass sculpture production. That support could come, in the way of collaborators rather than financial support. And we see our overall scientific partnership and exhibition as contributing to a larger emergent art and science ecology. And we are committed to carrying this ambitious project forward. We hope that you will help us continue our community building work between art and science, and please reach out to us if you're interested in working with us or learning more. Thank you so much.
Thank you.
Fantastic. Vet, Janna, anything here?
I have such an orthogonally weird question. And again, I'm projecting on all of you because these are things that are just on my mind, but I've been thinking about exoplanets, planets around other star systems and what it means for the emergence of life. And it's probably microbial life, right? That's what we should be looking for, because you can't even get near sentient life if you don't start with microbial life. And I wonder how our impressions of just how microbial life metabolizes systems and plant life and the very basics of the emergence of life in exoplanets, like how you can exploit that thinking to then look back at the Earth. Like, how different could it be? And how similar must it be? Like, what are the constraints and parameters that it must adhere to? And how does that inform our thinking about those kinds of fundamentals here on Earth?
Yeah, thank you so much for that thought. Yeah, we're very interested in, like you said, microbes are obviously sort of the earliest forms of life. They're the most prolific type of life on the planet. And the founder of Kingdom Supercultures, we've been talking to him about how much there is undiscovered and unknown about the ecologies between these microbes. So we don't really know how they might be recombined or how exactly their relationships unfold. So that's really what we want to analyze. And we would totally love to expand this research into some sort of space exploration. And we've definitely thought of before of how, our microbes come everywhere with us. So even as we're sending things into space, I'm sure there are some, slough off that happens. And, we just don't think about that because we never see these, or we rarely see these types of life. And it's so easy for humans who are so ocular centric to forget that that is the majority of life, and also of our own bodies.
Yeah, actually, we might well have put microbes on Mars. So we did.
And Perseverance has just landed, and it may well find our own microbes.
That's right. When we collect microbes from Mars, we're not going to know if we put them there in the first place.
Yes,
Nice.
exactly.
On that note, I am going to move us along to our last speaker for the evening. So thank you so much to everyone who's gone before, and for all of your energy and enthusiasm and passion that you've brought with you today, and all the work that you've just got into it. We've got our final speaker for today, which is Janet Biggs.
Hi.